Okay, let's do a quick review of protein structure in general. So we'll start with the amino acid, which is the individual subunit of the protein. And when we take a look at the generic amino acid image here, we notice that it's made out of 4 different elements. So we have carbon, hydrogen, oxygen, which is the same as carbohydrates, but then we also have nitrogen. Now, that basically makes up most amino acids. However, sometimes in the R group, we can also have sulfur. Okay, so there are some amino acids, there's 2 of them that might have a sulfur. So in terms of elements making up proteins, we have carbon, hydrogen, oxygen, nitrogen, and maybe sulfur. Okay? All right, let's move on. We already talked about these guys, so we can group these amino acids. By the way, there's 20 amino acids total. We can group them into nonpolar, polar, and charged. The nonpolar ones are going to not have any oxygens or nitrogens, or very few nitrogens, or they're sort of bracketed nitrogens that are bordered by carbons with very, very low or no polarity at all. So they tend to be hydrophobic. Okay, and they're going to engage later when we talk about protein structure some more. They're going to engage in hydrophobic interactions. So they like to be with each other, with other molecules that are hydrophobic or nonpolar. All right, I don't know if I ever talked about the names of these. So they're glycine, and the abbreviations are always like 3 letters or 1 letter. We're going to use the 3-letter ones. Glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. All right, so then the second group is the polar ones. They do have some kind of electronegative element, like oxygen, like nitrogen, like possibly sulfur. The polar ones are going to engage in hydrogen bonding. So these guys do the hydrogen bonding to form more complex protein structures. Cysteine's sort of an exception because it's going to form covalent bonds with this sulfur. We'll talk about that in a little while. And then finally, we got the charged ones that form ionic bonds because they have a full plus and a full minus charge on their R group. All right, so these guys are aspartic acid, glutamic acid, lysine, arginine, and then histidine. All right, so the way amino acids link together is actually pretty similar to the way that carbohydrates link together, or the way that monosaccharides link. Each individual amino acid can link to another one through the process of dehydration synthesis. So once again, we look for that OH, we look for that hydrogen, and that's going to form the water, just like before, except now we don't have an oxygen in between. We form a carbon to nitrogen connection. That carbon to nitrogen connection we call a peptide bond. In the case of the carbohydrates, we called it a glycosidic linkage. Here we call it a peptide bond. So when you see a long chain of amino acids, if you see the structure in a picture, you should be able to identify the peptide bonds between the carbon and the nitrogen. Now there's another carbon-nitrogen bond here, covalent bond, but that's not the peptide bond because that's just the bond that holds together this amino acid, this one. All right. So they can break apart through hydrolysis by just simply adding a water where the OH goes on the one side to form the carboxyl again, and then the hydrogen goes to the other side to form the amine over here. So basically just the reverse happens. All right, so let's talk about protein structure in sort of a larger picture. Proteins, as you know, can fold into very complex shapes, and there's sort of a hierarchy of the levels of structure going from primary all the way to a 4th level called quaternary. And primary is the simplest. It's just simply it's just the chain of amino acids. It's held together with covalent bonds, the peptide bonds, between the amino acids. So in between each one of these, there's a covalent bond, that peptide bond that holds them together. All right, so it's just the straight chain, or might be a little curvy chain or whatever, but no real folding yet in the primary, at the primary level. Okay, secondary structure then the primary chain, so that the amino acid chain is either going to form a helix, which is shown here, but it's kind of hard to visualize because they actually put all the elements in there, which is kind of impressive. Whoever made this picture is pretty skilled. I wouldn't be able to do that. Or they're going to form a beta sheet, and a beta sheet just simply takes the chain and just folds it back upon itself to make this flat kind of a sheet. And then what's going to happen is hydrogen bonds are going to form in between the different rows to strengthen that. So that's the beta pleated sheet. Pleated just means it has these sort of zigzaggy kind of arrangements. But a lot of times people just call it beta sheet. Just it's shorter to say that. And then alpha helix. And alpha helices obviously also have hydrogen bonds between the amino acids here to strengthen that and give it a little bit more support. So that's secondary structure. So let's take a look at secondary structure in a little bit more detail. Let's actually try to draw, we're not going to try to draw an alpha helix, but we're going to try to draw a beta sheet. So we'll just start by drawing some amino acids. You should memorize the structure of an amino acid. It's not super hard. It's got an amine over here. It's got a carboxyl over here, like this. And what I'm going to do is immediately assume that dehydration synthesis took place already, and I'm going to connect another one. So I'm going to put the R group up here just to flip it around a little bit and put the hydrogen here. Actually, let me undo that. Let's discard that. All right, I'm starting over. A good teacher would have edited this video now and cut this out, but I'm just going to leave it in. All right, so let's try that again. This time, if I make a mistake, you can make fun of me. All right, I guess we're going to red. All right, let's try it again. We got N, we got C, we got C. I said we're going to put the R group up here. I did want that, but then I have to put the double bond O down here, so that's the mistake I made, and the hydrogen goes down here. So that extra little hydrogen on the central carbon goes here, and then what we have over here is then the next one lining up, N, C, C again, and we'll put another one in right away, N, C, C. Now, if this R group goes up, the next one has to go down, so they flip-flop, and then the next one has to go up. That's the way we're going to do that. That also means that this hydrogen is going to go up, and that this double bond O is going to go up. Okay, so anytime the R group faces down, then the hydrogen goes up, and the double bond O goes up. And then the reverse over here again, where the hydrogen goes down, and this guy, and then the double bond O. All right, so now this is going to curve around over here, and I can't draw it, but it's going to come around this way and then back again. So as it comes back again, where the nitrogens were over here on the left side, now the nitrogens are going to be over here on this side. Okay, so now it's going to be NCC in this direction because now it's going this way. Over here it was going this way. All right, coming around, it's looping around over here where we can't see it. This one has a hydrogen going up, this guy goes up, this double bond O goes up, and then this R group goes the opposite way. So now you just do it again, and you line up this, notice how I lined up this carbon here with this nitrogen. I'm going to do it again. I'm going to put this nitrogen right below this carbon. I'm going to go C, C, and then this time the R group goes this way, and then the H goes this way, the double bond O goes this way, la la la. By the way, I'll post pictures of like a nice image of this that looks a little bit neater, but I just wanted to kind of show you how I created and how I do that. Notice nitrogen right below the carbon, because this double bond O goes down, this one's going to go up. Oh, the other reasoning for it is that because this R group was going up, the next R group has to go down. Okay, well, if that R group goes down, then the other guys have to go up. Double bond O goes up, hydrogen goes up, and this guy goes here. And this just keeps going in this direction. It just keeps going basically for many amino acids. So now we got to wonder, where do the hydrogen bonds form? So I'm wondering if I can maybe change my color here. I think I can. Let's make it a beautiful blue. And we're going to take a look at where are there electronegative elements. Well, here's one. Nitrogen is somewhat electronegative. It's going to be slightly negative here. The hydrogen is going to be slightly positive. I should put parentheses around it because it's partial. The oxygen clearly is going to be quite a bit negative, but still partial. This is slightly positive. because this draws the electrons to it. This has had the electrons sort of taken away from it. So there's an attraction here between this positive and this negative. That's going to form a hydrogen bond right here between these two. Over here, nothing, because carbon and hydrogen, they're just like not electronegative. They don't do anything like in terms of pulling electrons. Here again, we got negative, we got positive. Oh, look at that. Nothing here because the R groups don't have any role in secondary structure. So in beta pleated sheets and in alpha helices, the R groups don't do anything. They're going to be in tertiary, which is the third level of structure. So we leave the R groups out of this. There's no hydrogen bonds here. Here again, slight positive, slight negative, slight negative, slight positive, bam, we got more hydrogen bonds forming. So if I ask you to circle or indicate the hydrogen bonds, you know, if you're going to circle them, I would say right there, these are the elements that are involved in forming the hydrogen bonds. It's the hydrogen on the amine or on the nitrogen and the double bond O. It's technically not an amine anymore because it has this hydrogen removed. And this is technically not a carboxyl anymore because it had the OH removed when these link together, but you know, no big deal. So now we've drawn our first beta pleated sheet. So that's something you might want to practice so you get good at it in case I ever hold you to this. All right, so start practicing drawing these and getting good at it, and should be able to draw out like one with 6 or 9 or you know whatever, so many amino acids, and show where the hydrogen bonds line up. And it helps to have them all sort of on top of each other. Okie doke. Now we are not going to draw alpha helices. That we're not going to try. We're going to stick to beta sheets. In the alpha helices, we'll just trust that that's what happens with those. All right, we'll do tertiary in the next video. We'll end here.